Good morning, students. Today we will take the topic the fertilization comes under reproductive biology of angel sperms and plant morphogenesis. Fertilization is the process of the fusion of male and female gamete. In angel sperm, the female gamete is situated in the ovule, which is present in the ovary. Pollination could have brought in the male gametes present in the pollen grains on the stigmatic surface. The entire process of fertilization may be studied under the following steps. The first step is pollen germination and development of the pollen tube. Second, entry of the pollen tube into the ovule. Third point, structure of the female gametophyte. Entry of the pollen tube into the embryo sac. Fifth point, discharge of pollen tube contents. Sixth point, syngamy. Seventh point, that is double fertilization. Pollen germination and development of the pollen tube. This diagram shows in the pollen grain is a double layered structure. It consists of outer exine, inner intine, and also it bears the two nucleus, that is vegetative nucleus and the generative nucleus, or in other words, we also call it vegetative cell and the generative cell. Now, when the pollen grain fall on the stigmatic surface, the outer exine breaks open and the inner intine develops into a small tube-like structure. The vegetative cell develops into a tube and the generative cell gradually enters into the pollen tube along with the cytoplasm. Now, the generative cell, in the second particular diagram, the generative cell further undergoing a division, it forms two male gametes. One male gamete is larger in size, another male gamete is smaller in size. The larger one fuses with the secondary nucleus, the smaller one fuses with the egg nucleus. In smaller male gamete, the number of mitochondria is more. The vegetative nucleus develops into a tube. Second point, entry of the pollen tube into the ovule. The entry of the pollen tube into the ovule are of three types: porogamy, mesogamy, chelazogamy. The first one, A, that is, if the pollen tube directly enter through the micropyle, then it is called the porogamy. The second one, that is the mesogamy, if the pollen tube directly enter through the integuments, pierce through the integuments and touch the embryo sac, we call it the mesogamy. The third type, the chelazogamy, that is the entry of the pollen tube through the chelaza. Whatever may be the entry of the pollen tube into the ovule, ultimately the tip of the pollen tube goes up to the micropyle. Third one, the structure of the female gametophyte. The female gametophyte, we also call it the embryo sac. The embryo sac is a 7-celled 8-nucleate structure. Now, there are 8 nuclei are present. Out of these 8, 3 nuclei moving towards the micropylar side, another 3 nuclei moving towards the chelazal side. The 3 nuclei which are moving towards the micropylar side develops into an egg apparatus, and the another 3 nuclei moving towards the chelazal side develops into antipolar cells. The main function of the antipolar cell is to provide the nutrition for the developing embryo. The remaining 2 polar nuclei, one from the chelazal side, another one from the micropylar side, they fuse together to form a secondary nucleus. So this the diagram indicates that the antipolar cells are found towards the chelazal side and the egg apparatus are found towards the micropylar side. There, in the what we call the synagogues, the egg apparatus consisting of the egg in the center. On either side of the egg, there are 2 synagogues are there. The synagogues are face the filiferous apparatus. These filiferous apparatus helpful or facilitate the pollen tube into the embryo sac. Entry of the pollen tube into the embryo sac. This is the fourth point. The entry of the pollen tube into the embryo sac. In the first, the third one, there is the entry of the pollen tube into the ovule. Inside the ovule, the embryo sac is there. And the fourth point is entry of the pollen tube into the embryo sac. There are 3 types: between the egg and synapse, Another one between the embryo sac wall and one synergy, directly through one synergy. Ultimately, the accepted PV is always the pollen tube pierces through one synergy. When it pierces into the synergy, the piriforous tissues will secrete the chemicals and burst the tip of the pollen tube. Discharge of the pollen tube content. As soon as the pollen tube enters into the embryo sac, the tip of the pollen tube bursts, breaks open, and releases the vegetative nucleus and two male gametes. The vegetative nucleus gradually disintegrate and disappear inside the embryo sac. The remaining two male nucleus, one is larger male gamete, another is the smaller male gamete. The larger male gamete, less mitochondria is present. In a smaller male gamete, more number of mitochondria is present. Syngamy. The last point, that is the double fertilization. So double fertilization is a unique to angiosperms, as it is not found in any other group of the plants. During double fertilization, the second male gamete fuses with the secondary nucleus. Since the secondary nucleus is already diploid because the polar nuclei fuse together to form a diploid nucleus. Before fertilization, the embryo sac itself is a diploid. In addition to the diploid nucleus, another one male gamete fuses with the diploid nucleus, develops into a double fertilization. The fusion of the second male gamete with the secondary nucleus results diploid or triple fusion. The diploid product is called as the pen cell. Pen means primary endosperm nucleus. The primary endosperm nucleus divides and produces the endosperm, which nourishes the developing embryo. The formation of pen, that is primary endosperm nucleus, is said to be due to the double fertilization. The double fertilization product is two polar nuclei as one male gamete. So overall, if the fertilization completes, these 7 steps are very essential to complete know about the fertilization of angiosperm. But double fertilization is not found in any other plant except in angiosperm.